Hello and a very good day everyone. We are from Group 4. This is our group members. Today we are going to present about cDNA microarray and real-time PCR using Scorpion Primer. First and foremost, we need to understand the steps involved in cDNA microarray, which are sample collections, isolations of mRNA, creations of labeled cDNA, hybridization, collections, and analysis. For this question, the samples are collected from any part of Taxus brevifolia as an experimental sample, while the control sample is synthesized from the NCBI database. Second, the RNA is extracted by using PCI, and mRNA is isolated from RNA. Next, cDNA are prepared by using reverse transcriptase and labeled with two different fluorescent dyes and hybridized to the microarray. Dual-channel laser excitation excite the corresponding dyes. The spots may appear as green, red, and black, or sometimes yellow, depending on the expression level. So what should we do next? Do we need to do calculation? Do we need to put it under special microscopes? So how to confirm our result? Well, it is actually simple. If the expression of the particular gene is higher in the experimental sample than in reference sample, then the corresponding spot on the microarray will appear red. In contrast, if the expressions in the experimental samples is lower than in the reference samples, then the spots may appear green. Finally, if there is equal expressions in the two samples, then the spots may appear yellow. This slide here is just to summarize the steps in cDNA microarray. So I think that's all for me. Thank you.
To profile the gene expression of some of the identified candidate genes involved in the production of taxol, real-time polymerase chain reaction (qPCR) was performed, and this method allows the selective amplification of complementary DNA, or known as cDNA. To conduct real-time polymerase chain reaction, first cDNA was produced through reverse transcription, then the cDNA along with buffer, DNTPs, DNA polymerase, and primers were put in a tube. Then the tube was run on a real-time PCR machine. To quantify our expression, a detection method was considered, which we will be using Scorpion Primer. The Scorpion Primer gives a strong signal and acts much faster than other detection methods. Apart from that, since enzymatic cleavage is not required in this detection method, the time needed for signaling can be reduced. The unimolecular rearrangement mechanism of this method also allows for high sensitivity and extremely fast kinetic reaction. However, the downside is that this method is more laborious and expensive as it contains linker, fluorophore, and quencher.
So we move to the differences between the result obtained by quantitative PCR and also normal PCR. For quantitative PCR, the product can be detected in each of amplification cycle, and the data is collected during the exponential phase of reaction. While for the normal PCR, the product can only be detected by agarose jet electrophoresis, and the data is collected at the end of the reaction. For the quantitative PCR, the product is stained with fluorescent dye, while for the normal PCR, the product is stained with ethidium bromide. Quantitative PCR also allows the detection, characterization, and also quantification of nucleic acid, while for the normal PCR, it only allows analysis of a short sequence of DNA by amplifying a selected segment of DNA. For question number 1, the Scorpion probe is similar to molecular beacon in that it consists of a stem loop structure when it is in the free form. For the question number 2, so basically Scorpion primer is a bifunctional molecule in which a primer is covalently linked to the probe, and the molecule also contains fluorophore and also a quencher.